The last session was actually a perfect lead into what I'm going to try to talk about today. And I'm going to begin by reiterating what you heard from Karen about heterogeneity, which is, as we all know, there are many subtypes of ovarian cancer. Not a single subtype can really encompass everything we know about this disease. And the most common is a high-grade serous carcinoma. As you also heard, it's also the type that's associated with mutations in BRCA1. And it's the only one that was actually TCGA'd, right? So what did we learn from the TCGA? So this you saw from Doug. About half of all high-grade serous carcinomas are thought to have some defect in homologous recombination. A big chunk of that, obviously, is our knowledge about the BRCA1 and 2 genes, both germline somatic mutations, and we also talked a little bit about the methylation data with regards to BRCA1. And the thought is that defects in HR makes these tumors susceptible to DNA-damaging agents like carbo and cisplatin and PARP inhibition, which we will learn a little bit more about later in the session. And then there's the other half of the story, right, which are tumors that are not as well characterized. One of the genes in that other half is cyclinE, which is what I'm going to focus on today. And cyclinE is mutually exclusive with BRCA mutations. You never see the two together. And I'll talk a little bit about why that is later on. Now, since the TCGA, as Doug mentioned, we've learned a little bit more about the rest of the pie chart here, the 31% of other. David Botel's group had a very nice paper in Nature earlier this year where they were able to show with whole genome sequencing that there are gene breakage events that account for a big chunk of that 31 other percent. And that's gene breakage events in both the NF1 and RB genes. So now we know a little bit more about the full pie chart with regards to the genetic alterations that really are the hallmarks of this disease. Now, as I mentioned, I'm going to talk about cyclinE. And cyclinE works to really govern transition through the cell cycle by partnering with its kinase partner CDK2. And what overexpression of cyclinE oftentimes leads to is what we call unscheduled S phase. It pushes the cell into S phase before it's ready to do so. It creates replicative stress, and sometimes it's associated with centrosome amplification. Now, we were interested in addressing the role of cyclinE by going back to what we knew with regards to pathogenesis, which was that, as you heard from a number of speakers, we now believe that at least a majority of high-grade serous cancers arise from the fallopian tube, in particular the fimbriated end of the fallopian tube, where we've been able to identify precursor lesions for high-grade serous carcinoma. And this is just a summary slide of a lot of data that's been published by our groups and others, really sort of highlighting the two precursor lesions, a p53 signature, which is really benign appearing secretory cells that have mutations in p53, stabilized p53 protein, evidence of DNA damage as measured by the H2AX marker, and then the intraepithelial carcinoma, or the serous tubal intraepithelial carcinoma stick, which has all the hallmarks, cytological hallmarks of cancer, but has retained the features of p53 mutations, DNA damage, and this is the invasive cancer, which is what we know most about in terms of genetics. So we decided, let's go back and see how early is cyclinE deregulated in this progression model. And so this is work from Alison Karst in the lab, who was able to obtain, fortunately, a number of samples where we could look at some of these precursor lesions. And so we started by asking, if we look at p53 signatures, which are really defined by benign-looking, non-proliferative epithelium, which is what I'm pointing to with my arrow here, that's stained very strongly for p53, but don't proliferate, we see that there's high expression of cyclinE in some of these precursor lesions, and about a third of the ones that we looked at. When we looked at stick lesions, which are obviously intraepithelial carcinomas, about 2/3 had upregulation of cyclinE, suggesting that cyclinE potentially deregulation occurs early in this process. And so we thought we'd be able to model this by going back to our bucket of model systems that we've developed over the last 5 to 10 years, really aiming at trying to understand what's the fundamental susceptibility of the fallopian tube epithelium and how can we best study that? And this really is a slide illustrating the 4 major models we've looked at, where we can, for instance, in model 1, we can look at benign epithelium and its susceptibility to transformation. We can also look at the secretome using a model like this, and we've been doing that for biomarkers for early detection. We've used the fallopian tube secretory cell line to really study the effect of any given genetic alteration on the ability to transform these cells. We've generated genetically engineered mouse models that target the murine fallopian tube Pax8 positive cells, and we also have a bank of tumor avatars or PDXs that help us look at therapeutic opportunities in this area. So to look at cyclinE, we went to our cell line model where we were able to generate primary fallopian tube secretory cells from fimbrias obtained at the time of surgery. We immortalized these cells with telomerase and a mutant P53, which is one of the more common mutations, R175H. And then we either overexpress cyclinE or an empty vector control. And so when Allison analyzed these in culture, and I'll just go through these a little bit slowly, in panel A, we see that, as expected, there are higher levels of cyclinE in these cyclinE-expressing cells. This leads to activation of CDK2, its partner, which makes sense. It's now activating the complex, pushing the cells into S phase, as I mentioned. As a result, these cells grow faster. They're clonogenic when you see them at low density. They lose contact inhibition, which is very similar and consistent with the old work from Steve Reed in 1999, where he published the effects of cyclinE overexpression in NIH3T3 cells. And then they're able to form, though very weakly, colonies in soft agar, but really nothing robust like with a true oncogene like MYC or RAS. Now, what we did find was that when these cells are pushed into S phase so they can proliferate faster, we noticed that there was an increase in DNA damage. And this was measured by looking at phosphorylation of H2AX. So here's an increase in H2AX foci in the nuclei of cells that overexpress cyclinE, and this is quantified on the right. And if you look at a more generic form of DNA damage using a COMET assay, which really measures the fragmentation of DNA in any given cell, we saw that there was also an increase in COMET tails, which is fragmented DNA, in cyclinE overexpressing cells. So really, by all, this was consistent with prior work. What was really exciting for us was when we profiled what's happening in these cells compared to the empty vector control. And so what you're looking at here is a volcano plot of genes that are upregulated in cyclinE overexpressing cells versus genes that are downregulated. And what we noticed is that a lot of the genes that are upregulated in the cyclinE overexpressing cells are DNA repair factors that are involved in replication fork protection. Kind of makes sense. You're forcing the cell to go into S phase. It's causing replicative stress. In order to tolerate that stress, the DNA repair machinery is being upregulated. And we were able to validate this biochemically by showing that, as expected, cyclinE overexpressing cells have increased DNA damage. Some of the same genes that we found in our profile were biochemically validated, like XRCC2 and FANCD2. And when we went back to the TCGA data, we asked what pathways are associated with cyclinE overexpression, and not surprisingly, the Fanconi and homologous recombination pathway were among the top pathways associated with cyclinE amplified tumors. But the really exciting part about the observation was when we went back to David Botel's study that was published in the same year, where he did a synthetic lethal screen using RNAi, so to knock down all the genes in the genome and ask, in a cyclinE amplified cell, which genes, when I knock them down, will kill these cells? And the degree of overlap between what we found in our profiling and what they found in their screen is unbelievably high. Almost every single gene that we found upregulated as a result of cyclinE overexpression was also synthetic lethal, suggesting that cyclinE not only creates an upregulation of DNA repair factors, but also a dependency of these factors for these cells to survive. So, to Noah's point, which is how do we target amplification? cyclinE is one of the more abundant and common amplifications. Also mentioned earlier is the fact that it's associated with poor outcome and platinum resistance clinically. So this begs the question, how do we go after this? Now, one of the things we noticed was that one of the genes that was upregulated is XRCC2. And we heard a lot about the RAD51 paralogs this morning, RAD51B, RAD51C, RAD51D. Well, it turns out that those three genes and XRCC2 form a complex called the RAD51 paralog complex that helps the RAD51 filament become more flexible so that it can actually conduct homologous recombination. And we find that it's pretty interesting that cyclinE overexpressing cells upregulate this pathway, suggesting that there's a dependency in the paralogs. When we actually looked at the TCGA data, we saw that there's a very strong correlation between cyclinE amplification and XRCC2 levels. But even more striking, when we look at pan-cancer analysis, we find an even stronger correlation between cyclinE amplifications and the overexpression of this RAD51 paralog XRCC2. Now, I don't have time to get into some of the work we're doing right now, but we've been able to map the interactions between XRCC2 and some of the other members. And it turns out that if you interfere with the binding domains of how that complex gets assembled, it effectively does the same thing as knocking out XRCC2. So it's actually a synthetic lethal event, and that may be a therapeutic opportunity. And this just shows us that if we take an amplified cyclinE cell, a cell with amplification cyclinE, versus one that doesn't have the amplification, and we knock down XRCC2, we see that it's lethal in the amplified cells and not in the cells that don't have the amplification. Again, suggesting that this may be an opportunity to go after these tumors that have these amplifications. And again, this is a paper that just came out this year where in C. elegans, they looked at what's the function of these paralogs, right? Because it's very poorly studied. And again, it seems that they are able to bind the Rad51 filament and cause it to be flexible so that the filament can now cause strand invasion and really mediate homologous recombination. In the absence of these paralogs, Rad51 filament cannot invade and actually execute HR. So it seems one opportunity is that cyclinE overexpressing cells have a tremendous dependency on DNA repair. So unlike the BRCA side of the equation, where the DNA repair pathway is defective, on this side of the equation, the DNA repair pathway may actually be a therapeutic target. Okay? But we also know that the most obvious target to go after with regard to cyclinE, it's its kinase partner, CDK2. CDK2 itself has been developed as a therapeutic target in work that we've done with David Botel. When you take these amplified cells, the cyclinE amplified cells, and knock down either cyclinE or CDK2, we see that there's a tremendous decrease in proliferation in these cells. But cells that don't have the amplification really don't care. So it seems to be functionally as if cyclinE is a biomarker for tumors that may be susceptible to CDK2 inhibition. Now, there are a lot of CDK2 inhibitors in the clinic. We've learned a lot about the CDK4 and 6 success in breast cancer. CDK1, 2 inhibitors do exist. Dinucyclib, developed by Merck and a few others. The problem with most of these is that it never hits just 1 and 2. It hits 9, it hits 7, causes a lot of toxicity that has been sort of a hindrance in the clinic. The other problem is that in most studies where it's been used across a population of patients without really selecting out those that have a cyclinE amplification. And we would predict that the cyclinE amplified tumors, regardless of what histology and where they come from, would be the ones that would be susceptible to CDK2 inhibition. And just to give you an idea of how we've tried to address this potential therapeutic opportunity, you'll remember that cyclinE by itself was really not able to transform the cells, the fallopian tube cells, in culture, right? It caused them to proliferate faster. It caused some DNA damage, but it really wasn't able to enable them to grow under anchorage-independent conditions. And so we thought, well, maybe you need something else that will cooperate with cyclinE. And so we looked, instead of doing a whole genome gain-of-function screen, we actually looked at all the genes that are amplified in the recurrent amplifications of high-grade serous ovarian cancer. There's about 63 recurrent amplicons across the genome. There are about a little over 1,200 genes that are in those amplicons. And so working with Bill Hahn and the Broad Institute, we made a gain-of-function ORF library, open reading frame library, where we introduced these genes into the cyclinE overexpressing cells and asked which one of these could cooperate. And we did this by measuring their ability to form colonies, anchorage-independent colonies, both in soft agar and in ultra-low attachment plates. And what we find here are the results. There were a number of hits. All in all, there was some overlap between these two assays. And a number of the targets, the cooperating genes here, are also synthetic lethal with cyclinE, which is also an amazing result. Right? Some of the again, same things that cooperate for transformation create a dependency on those same genes. One of the genes we heard about earlier in terms of endometrial cancer is AKT itself, and AKT1 and 3 actually came out in this screen. So we've actually done a study now that we're in the process of writing up, so I'm not going to get into too much detail, but it turns out if you reduce the dose of dinucyclib, right, because I told you it was a toxic drug, right, and now do a synthetic compound library screen, looking for things that will synergize with dinucyclib at doses that aren't toxic, a number of things came out, but at the top of the list was an AKT inhibitor. So it turns out that AKT not only cooperates with cyclinE for transformation, but it may also be a sort of therapeutic opportunity as well. And so in studies that were published by the Sanger, in OVCAR3, which are cyclinE amplified cells, one of the drugs that was most effective was AKT inhibition, consistent with some of the things we've seen, and we've also done this in vivo. So it suggests that cyclinE amplification, which accounts for 20% of all high-grade serous tumors, may actually be a druggable amplicon, right? The opportunity is to go after its partner CDK2. The opportunities are to go after the DNA repair system that it becomes dependent in that setting, right? Because if you get rid of them, it causes genomic catastrophe and the cells die. So there's at least two opportunities to go after an amplification in high-grade serous ovarian cancer, but you have to sort of know where to look for it, right? You can't use these drugs necessarily in all comers. It would have to be with cyclinE as the biomarker for this cancer. So in summary, we've heard a lot about the genomic and genetic heterogeneity in high-grade serous carcinoma. cyclinE amplification is one of those amplifications that's kind of a hallmark of this disease, mutually exclusive with BRCA1. We now know why it's mutually exclusive with BRCA1, because they are synthetic lethal. They are in the same sort of pathway. They create a dependency on each other. cyclinE amplification in high-grade serous carcinomas depend on this replication fork protection pathway, and that may be a therapeutic opportunity. And again, CDK2 inhibition in the right setting with cyclinE as the biomarker may be also an opportunity for us to go after this particular form of high-grade serous cancer. And being a pathologist, I will conclude with my acknowledgments in a pathologic way. Some of my colleagues back at the Dana-Farber and my new colleagues and friends at the University of Pennsylvania. Thank you.